Okay, today we are talking just about the mapping and just visualizing the mapping of the RNA-seq against the reference genome. And but before starting that, just please be aware that download the RStudio for our Monday for next session that we are just doing the expression analysis. Bring that. We are just starting the. Okay. Let's just go to our ACF account with this one. Okay. And then to the directory of your directory in the home course section. And then you just need to create few directories for getting the genome annotation, the reference genome, and also raw reads. So this one, let's say, go to here. Encoded. I think it's the same. Yep, RNA-seq lab 1. I think on the upper part, I just created another directory because I didn't want to delete the rest of the files. That's just put the files exactly in the course website.
Do they'll have a user name folder and then what people should get in that? Does it matter?
No, it doesn't. It should be RNA-seq lab 1. The rest is the same. But it's not the same in the directory that was created. And then just go to the create another one for the raw data. And then go to the raw data and get the reference genome of the RNA-seq reads that in this course we are talking for this lab, we are using the Arabidopsis polyana reference genome. And we will get it row wget. How can I paste it? Oh, sorry.
Yeah, you'll have to recopy it.
It's all.
Let's see.
Oh, and the next one that the file we need is the annotation, genome annotation of the Arabidopsis again. And we unzip all of them, decompress them with gunzip -gz, gunzip. And these two links just show you how the structure of the files are, which the study that we chose today is 16 paired end reads for this Arabidopsis thaliana under 4 different environments and with 4 different phenotypes. And the reads that you need to download actually are the ones in here as a FASTQ files FTP that since it's about 32 reads, so I just downloaded 2 of them as a 1 third end reads, and we are doing the analysis today based on one of these reads, and then at the end I will show you how to do a loop command when you get all of these reads and you want to do all of the mapping against all of these paired end reads for this study. And at the same time, there is another link that shows the structure of the data, which shows which read belongs to which phenotype and which in treatment. Let's say, for example, the one I'm going to use is this one, which ends by 140. This is this phenotype and is under the treatment of dehydration. So we are going to download this one. Actually, you are not downloading because it takes maybe 5 to 10 minutes. You will create a soft link from my directory that I've already downloaded this, and we use this only read for the rest of our mapping, alignment, and also visualization through the IGB today. So go back to the. So let's start just by getting an interactive session.
We're not using it for anything like that, but it's small.
And when I tried it with the processor per node 1, the mapping takes about 20 to 21 minutes. And when you get or ask a whole node, I don't know how many processors in that node, it takes maybe about 6 to 7 minutes per read to be mapped against the reference genome that we have. So in here, we're just asking for 1 processor per node, which will take about that 20 minutes, but at the same time, we will do other things just to save our time. So, I don't know how much should I wait. Yes.
Can you tell us anything about star while we wait?
Oh, sure. For the star, first of all, we should index the reference genome to have a background or database for the software to start the analysis and compare the reads against the reference genome. So the parameters that in here is used is the run mode is kind of a parameter in the valid in the software that just run genome indices to create a database for the Next step, which is alignment. The genome dir, that it's just a folder that you create. You have to manually create it to put the genome indices in that, which is in here, genome dir. And the next parameter is your raw reads. Is your genome reference genome, which shows by genome FASTA files. And then the number of the processors, which is in here, is exactly one node, one processor per node. The next one is GTF file, which just shows the annotation of the file. In this case, we use the Arabidopsis annotation with GTF file. And the last one shows the length of the genomic sequence around the annotation of the annotation junctions, or the portions of the genome that is splicing sites, and shows which parts belong to which region that code for sequences. And the important things for that, the GTF file, is that you have to specify a length spanning between these splicing sites, which shows the read length, which needs the read length. And in this case, we use a read length -1, which you have to get the read length of the read 1 actually, and read 2 is a paired end read. And you get the one that has the higher higher or more read length. For example, in our case, the read number 1, I will show it to you after we get this interaction, the interactive node done. The read 1 has 100 base pair, and the read 2 has 102. So we just get the bigger one and do it subtracted by 1 and put it in the parameter of the reference genome, parameter of the index creating by the software. So I'm still waiting for this.
This job is actually long.
So I will just go ahead and continue the rest. It's not a good thing to just run it on the head node, right? Okay. For the next thing, after we get the indices done, we have the background, we have the database to start the mapping. We need our raw reads and our annotation, and also the genome indices that we just created. So we just start with the parameter genome dir, which is specify the genome indices were located, read files in the raw reads, which is 1 and 2 actually is a paired end read. So to just specify the read number 1 and number 2, and then you just define where to put the output files, and also the format, in the format of the alignment, which is in here, BAM, which is defined as a BAM file because we need it for the IGV, for the visualization, and it must be sorted BAM file. So we define a sorted BAM coordinate file, and then again, like the indices creation, we just define the number of the nodes that again we are using. This is again 1. It's not happening anything. And since in this study that for 16 reads that we have, it's too much. I think it took about 2 days for me because to get the alignments done, because every time the jobs were killed, I couldn't run them at the same time. So for the days that you have your own data set or you want to work on this project, to have this works done, instead of doing 1 by 1 reads, doing through the software star, this would be actually this command is a loop command for all reads, whatever reads you have, 100, 200, or even 10, the even shorter number of the reads, which just as a brief description for this one, the first line just get the names of the reads, and then goes to the second, which just you ask the command just start running based on the for loop command, which starts by replacing the names of the first read to the second read, which is, for example, let's say DRR and 100 -1. It just remove 1 and replace it with 2 to just facilitate your works instead of doing 1 by 1. And then this echo, it just shows this reads are processing, and then goes to the star, which is exactly the same that I just previously explained about it in the software, that how the stars works. And then you have all of your reads in the alignment output. And here we need it.
If you want to just do it on the head node, and everybody whose job isn't launched, like, don't run the head node. If your job is launched, go for it. But if it hasn't, maybe just don't do it. Once it launches, we can go back and go through some of this. But normally, if you just want to,
sure,
we'll see that keeps up and move on. But it doesn't look very promising.
Okay. And in here in the raw data, I just create a soft link from the data that I downloaded, the raw data, the perp and raw data, just by LNS and the pathway that is the raw data are located. Okay, this is the folder contains the raw data. So now we have all the files for creating the indices through by the reference or index the reference genome of the Arabidopsis. So we have to just first module the software, which is this one, modulo star. And if you type star, you will get all the manuals and the stuff that shows how the software works. Okay, we type star. And okay, just before doing that, it's better to create some other folders. Encladir. And the right tab. Okay. It's for our analysis. We need to go into it and copy.
So that double ampersand is not something we've seen in this class before. You want to talk about that?
Yeah, when you want to just, instead of doing one folder and then hit enter and then that again double type it, instead of doing multiple things in command, you have one line and do all of them at one command. Just create it and then go inside it, and then you can also have another and enter to the, for example, the next folder, the next folder, and next folder. It is in the analysis. It's in the analysis that
analysis and the line star.
Right.
And just pretend that that's two different things.
We can have it like, okay. In our RNA-seq lab, we just created the analysis and raw data. In the raw data, we have our genome annotation, the reference genome, and the raw reads. And then in the analysis, we have our star. Maybe somebody likes to have maybe another analysis, just another folder for, let's say, another software, which could be HiSat, which could be RapMap, whatever they like. So they can create another folders for that. That's the reason I just create this one, just to be a bit consistent with maybe you want to check.
You need to add CD analysis after make your analysis. I think if we want alignment star and
Make the alignment, CD alignment. I think so. Yep. Right. Sorry about that. And then the last one that we need to create is the genome dir, which is required by the software to for putting the genome indices inside that, which is inside the alignment star. Okay, and from here we just do not need to. Oh, sorry. We do not need to go inside the genome dir. We just run the analysis from the alignment star folder. Let me just delete this folder because it should be okay. Okay. So on star, the first parameter is the run mode, which we define what the star do. Actually, it's just generating the genome indices. Genome generate. And the second parameter, the genome dir, is exactly the current location that we just created, the current directory we created, genome dir. And then the file that, the folder that contains the reference genome, FASTA file. So it is in here, the raw data, Arabidopsis, DNA. And then we define the number of the processor, which I don't know, just put 1 in this case. The next parameter is the annotation, the genome annotation file. Okay. And then just enter. Depending on the processor that it gives, between 7 minutes to 20 minutes. If you have one node, one processor per node, 20 minutes. The whole processor, whole node, 7 minutes only. Just gonna show you how to identify the number of the read length, how to get it. Just now I can feel how slow logging is now. It takes 10 seconds.
That's probably us.
It's a bit weird, but okay. Okay, just for clarification of how to identify the read length, just get the head and the tail of the reads. So AWK, which is a bit which is not AWK. Okay, the first one, it's just showing the number of the reads per the variation from the smallest to the longest read length per the first read, DRR 16140 underscore 1, which is varied from 56 to 100, and the second read is 56 to 102. So we just choose 102 and just subtract it by 1 and put it in that place as the overhang value for doing this start, for the creating the reference, indices of a reference,
You can also just paste a read into like Word. The number of characters.
Okay.
There's like character counters on the internet where you can just paste a read and it'll tell you how many characters.
Yeah, that's also good.
I mean, this is nice. This is a nice fancy way to do it. But if you're not going to remember it, because I don't ever remember how you talk.
Okay, at the same time, since it's going, I don't know how long further mapping it will take based on we are in the head mode, but maybe we can copy the results that I have already done, and the students can copy it in their own directory. It's working. Okay. But since it's going, could you just go and copy the annotation file and the reference genome that we just downloaded from your directory to your local computer, because we need those two plus 2 other files for our visualization. Could you just please download them in your local computer since we are going with this indices? Which is exactly in the section of the website course visualize with IGB. Just find those files in your raw data that you just got, the GTF file and the .fa file. Copy them in your local computer. Okay, good job. We're finally done with the indices. Does it need to students copy the genome directory to the, if you have already get that interactive session, just copy the directory that I just created for the genome indices. And if not, you do not have that interactive session, just watch whatever is in here. I think it's better not to run it.
Here's what I think the other thing we can do. He can kind of show you, like here's going to show you how to do it on command line. Maybe I haven't written the homework for this week yet, so maybe like the first time I'm talking to you, like we'll give you a Q subscript and you can paste in the commands and run it. Because I think what's going to happen is, or not Q sub, yeah, like you can create the script and then just launch it and it'll run over the next few days whenever there's resources on the ETF. And then you'll have all the files in your directory by the time you get back on Monday. So I'll take that like the first one. So if you're feeling like, oh, I can't, I never got a Q sub login, this is confusing, you don't have to worry about it. You can try to copy all of this and get it on your computer and make it work. But we'll also get you a script to make sure that you can get the files to read in your own directories. And I will call and complain to the ACF as well. Hopefully we'll all be able to log in with it.
Okay, now we have our genome indices, which is located in the directory genome dir. And from here, we run the alignment with this command star. We define the genome indices folder, which is in here. And then the raw reads, which defined by this parameter. And we just define where they are located. It's not going on. There's nothing in that. Why doesn't he read the name of the files? It's already there.
Back to the beginning of all the
It's in raw data. There should.
Oh, you have raw data twice. The top one is the one you created, and the
Okay.
second one is the one you created. That's involved.
Yes. So just for the raw reads or the paired end reads after the double dot slash and raw data, add another raw data slash to get the raw reads actually.
You guys catch that? Like, you created a raw data, and then like it's symbolically created a raw data file. There's like a raw data, and then another folder underneath.
And then the second read, the next parameter define where the output file should be. The next parameter define that we need a spam sorted file. It's actually the output. Coordinate. And then number of the threads. Because we define it to 1. And then hit enter. Thank you. Alignment output. And I think the error in here, I did not create any output file, output directory for it. So I have to return back.
The good news it might be kind of confusing. Bouncing back and forth between the directory and the lesson. All the files actually exist, and the directory it keeps using to show you how to do it. They're like getting confused with different paths and different cells. But I think you can follow the directions on the website.
Yes, I didn't create it actually. It should be encoder alignment output. Alignment output. So we run it. Is the path. And out.
You created alignment output. Alignment out.
Yes. Yeah. Alignment out. So it gives you three things: starting, loading, and then the genome indices, and it takes maximum 20 to 25 minutes, 20 minutes, let's say. But at the same time, you can copy the results that I just created a few days ago, which you need to.
Well, it's 411.
Okay.
We can do it next time.
Next
You'll have the entire class for your next Wednesday.
Okay.
So I will post the first time more question will be something like, here is the kernel of the qsub command, like shell script, paste in the commands to run the genome generation. I'll figure out some way to show me. I'll try to have that posted tonight. Put it in my class. Do you guys have any questions? Sorry, I didn't mean to take. Go ahead.
No, I'm fine. I just
yeah,
I just wanted to say that we'll fix it, and I think there will be in the next session. I will continue just by everything set up, and I think there are some points in the website course for these directories. I will fix them, make them easier to follow up, and sorry about today. Thank you, guys.
I have this great book from a guy who like his whole thing is like teaching people how to teach computer science, especially the sciences. And he published this book, and on the front is like a little sign that one of his little kids wrote that says do not be worried and you will not make mistakes. And then it's like little hammer.
Yes.
Right? And I feel like it's just like whatever happens in the middle of the lesson, you just roll with it.
Just deal with
it's a learning experience for you guys.
Yeah, especially
I make this complicated on purpose because you guys need to learn. Like that's my general go-to.